Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. Today we are going to learn about dosage compensation. So in last class we learned about long non-coding RNA when we were discussing about non-coding RNA, the RNA which do not code for any protein. Today we are going to discuss about dosage compensation. So this phenomena, dosage compensation, is also epigenetically regulated. But before learning about the regulation of this, we are going to learn what is dosage compensation. So this is a mechanism by which the genes which are sex chromosome linked and those sex chromosomes differ in the number in the males and females of an organism. So the expression of such genes is equalized, and that phenomena is known as dosage compensation. For example, in humans, females have XX chromosomes, while males have XY6 chromosomes. Okay, on a diploid set of autosomes, right? Okay, so the females were so the X-linked gene products in females would be twice of that of the what can be expressed in males. Okay, and this can create imbalance in female cells. So for creating a balance, the number of the expression of such sex chromosomes which differ in males and females, that is equalized, and this equalization of the gene products or the expression of the genes is known as dosage compensation. So there are different mechanisms by which the expression levels are balanced. Let's take a look at the mechanisms. So in mammals, there is X inactivation, which does the dosage compensation. Means two copies in the females. Out of those two copies, one copy of X chromosome is inactivated. So now there will be only one functional X chromosome in females as well as males. So this creates a balance. Okay. There's other mechanism known as hyperactivation of X chromosome, and this is seen in Drosophila. Okay. So in Drosophila, the females have XX chromosomes while males have only single copy of X chromosome. The single copy of X chromosome in males, this is hyperactivated, and due to hyperactivation, it creates two times of the gene product. So this equalizes what is present in females, and this is how the balance is achieved. The third mechanism, which is seen in C. elegans, this is quite different from humans like mammals and from Drosophila. So in C. elegans, instead of hyperactivating a single chromosome, or instead of inactivating a single chromosome, the hypoactivation is seen, but in the both of the X chromosomes. So in hermaphrodites who have both sexual organs of males and females, those have two X chromosomes. So both of the X chromosomes, they are hypoactivated. So they are hypoactivated by 50%. So the gene products is decreased by 50%, and that is how the balance is achieved. And so now the only half of the product will be produced by both X chromosomes. So this equals, this balances, this equals with the males who have single X chromosome. Okay. So this is how there are different mechanisms. In mammals, one X copy is silenced. Okay. In the Drosophila, the one X chromosome in the males is hyperactivated, while in C. elegans, the both of the X chromosomes which are present in hermaphrodite, they are hypoactivated. So these are three different mechanisms by which dosage compensation is achieved. So we are going to discuss about X inactivation in detail. So let's learn how this X chromosome inactivation was first identified by researchers. So in 1949, two scientists, Murray Barr and Bertram, both of them observed something, and that was the first phenomena in the X inactivation history. So they observed, this is, let's assume this is female cell, which is an interphase, and this is the nucleus, and this is the male cell, and this is its nucleus. So when they observed those cells under the microscope, they saw that there is a densely stained structure in the female cell, which are in the interphase stage, while there is no such structure present in the male cells. So this was observed in all of the female cells. So this densely stained structure was termed as Barr body. At that point of time in 1949, this structure was named as Barr body. Then in 1959, there was a researcher named Ono. He identified this Barr body as inactivated X chromosome. So he observed that this densely stained Barr body is nothing else but the inactivated X chromosome, the X chromosome which is heterochromatinized, and that is why it is inactivated. Then in 1961, there was a researcher known as Lyon. She proposed a hypothesis which is called as Lyon hypothesis. So Mary Lyon proposed an hypothesis that the X chromosome is inactivated, right? One of the two X chromosome is inactivated, but the choice of the X chromosome which is going to be inactivated will be random. Okay, so she was the researcher who proposed random X chromosome inactivation. This is nowadays known as Lyon's law. Okay, so there is random X chromosome inactivation in earlier stages of the life cycle, earlier developmental stages of an organism. So this is a well-established fact nowadays. So this was how the X chromosome is inactivated in females, in humans, in mammals. This was established by researchers, and this was the history of the X chromosome inactivation. But there are certain disorders. Let's learn about the disorders that are linked when there something goes wrong in the dosage compensation. So disorders related to dosage compensation. The first one that we need to focus on is Turner syndrome. Okay, so in Turner syndrome in females, they lack one X chromosome. So instead of two X chromosomes, they have XO. So they have single X chromosomes, and that causes the Turner syndrome. Sometimes if something goes wrong in dosage compensation, it can also lead to lethality. There's other syndrome known as Klinefelter syndrome. here instead of XY chromosome in males, there are two X chromosomes in males. So genotypic same like I mean the genotype would be the 22 autosomes, the 22 pairs of autosomes, okay, and XXY chromosomes. This would be the genotype of the males. That is there was once in 1965 in the Olympics, the bar body was used to identify male and female athletes. So that was discarded later on because it could give false positive as well as false negative cases. So, for example, in Turner syndrome, the females who have Turner syndrome, they only have one X chromosome, right? So they will have zero bar bodies. Okay, so there will be no bar body and they will be declared as males. But in case they are females, so in such cases, the bar using bar body to identify males and females, this goes wrong. So there are other cases as well. For example, if there is triple XXX syndrome, so we have three X syndromes. That is why we will have two bar bodies, right? From this it will be ambiguous whether it is male or female, right? Again, in Klinefelter syndrome, the males who do not have any bar body, they will, due to presence of extra X chromosomes, they will have one bar body, and those males will be declared as females, right? And that can cause problems. Then again, there's tetraploid situation. Okay, so like we have four X chromosomes in females plus 88 autosomes. Okay, so we like instead of 2n, we have 4n. Okay, so the X chromosome, X inactivation is done per diploid set. Okay, okay. So when there are two n of the cell is 2n means it's diploid. It will have 44 autosomes plus two X chromosomes. In that case, one X chromosome will be inactivated. If the cell has 4n composition, it is tetraploid. It will have two sets of the diploid, two sets, so like 44 autosomes plus 2x, as well as again 44 autosomes plus 2x. So that is why instead of three bar bodies, like here we had three X chromosomes, so there were two bar bodies, but here we have four X chromosomes, so logically we should have three bar bodies. But in this case, the X inactivation is always per diploid set. Okay, it is so this X inactivation is done to maintain the sex chromosome to autosomal ratio. So it is always the X as to 2a ratio. Okay, so the diploid set of the autosomes. So that is why instead of three bar bodies, which is logically correct, this tetraploid condition will have two bar bodies. Okay, so I hope it is clear why, because from this state only one X will be inactivated, and from this state one X will be inactivated. So the final composition will be something like that: 88 autosomes plus two bar bodies, and yeah, obviously two X chromosomes. So this is how we can identify how many bar bodies will be present in a certain individual and what will be the genotype of that individual. So due to the false positive and negative cases in Klinefelter syndrome as well as in Turner syndrome, the idea of using bar body in the Olympics to identify males and females, this was discarded. So this was done only once in 1965, only in that year. So this is all about dosage compensation phenomena. In next lecture, we will learn about the X chromosome inactivation, the X chromosome inactivation center, XIC, and how it is epigenetically regulated. When does it happen? How, why, if it is random or imprinted, we will learn about the choice. We will learn about kiss and go mechanism. We will learn all of those things in the next lecture. Till then, see you. Bye bye.